Welcome to the next lecture of our course on environmental biotechnology. In this particular lecture, we will continue our discussion on bioremediation, and in particular in this lecture we will move forward from the pre-genomics approaches to the whole genome and then subsequently the metagenome based approaches adopted for bioremediation. Now, if we look at the early days of bioremediation research, as I discussed in my earlier lectures, that isolation and characterization of relevant microorganisms, particularly bacteria, were considered to be the most important step towards bioremediation. And as we are able to actually divided this course of development of bioremediation into at least into two phases, major phases. One is considered to be the pre-genomics phase or pre-genomics approach, both including the non-molecular as well as molecular approaches, and then post-genomics approaches. Here the term genomics basically refers to the whole genome sequencing and high throughput next generation sequencing based approaches, which also include the metagenomics and transcriptomics, metatranscriptomics, etc. So, in the pre-genomics phase, or when we were actually not doing much of the whole genome sequencing or the metagenome analysis of the samples in order to understand the bioremediation activities or in order to identify the organisms which could be useful for bioremediation. Major emphasis used to be given or is given still in some cases or some places, emphasis is given on isolation and characterization of microorganisms. Now, this isolation and characterization could be done by various ways, and the characterization particularly refers to the metabolic or ecophysiological characterization of the responsible organisms, which are identified to be the potential candidate organisms. So, here the word responsible refers to the potential candidate organisms, and subsequent to these characterization processes, we actually were able to identify the major metabolic activities or the catalytic activities of the particular organism or the candidate organism, which will make the particular organism suitable for its application in various bioremediation process development. And this not only includes the isolation and characterization of the organisms which are isolated, but also helped us to identify the kind of nutritional and or other amendments which may be required for the particular candidate microorganisms in order to survive well within the polluted or the contaminated environment, and those are considered to be the part of the engineered bioremediation. Now, during this process of time, the initial phases were mostly non-molecular because of the lack of the adequate molecular biology tools, but subsequently as the molecular approaches were available, like the PCR, cloning and sequencing were available. So, we were able to utilize the tremendous capacity of ribosomal RNA based identification of those isolates. So, the isolated bacterial taxa or the isolated organisms, their taxonomic identity were very definitely delineated, and it is not only the ribosomal RNA gene based taxonomy, but also different type of polyphasic taxonomy. Basically, that includes along with 16S ribosomal RNA, fatty acid methyl ester analysis, lipid marker, quinone and other single copy marker genes which are good for determining the taxonomic affiliation of the organisms were also included, and eventually the genetic analysis of the isolated strains were performed. And all these non-molecular to molecular analysis helped us to overcome some of the drawbacks of the non-molecular, so called non-molecular approaches, particularly the uncertainty about the importance of any particular isolate or isolates in their in situ performance, and about the subsequent fate of the contaminant in its modified form. So, these were actually identified to be the major drawbacks of the non-molecular techniques, but with the molecular techniques we are able to actually delineate that how this target organism, the candidate bacterial strain or strains which are isolated and developed and characterized in the laboratory were later exposed to the real environment, how they are going to be performing. We using the 16S ribosomal RNA techniques we are able to monitor that. It is also remain a point of concern about the fate of the contaminants in its real environment, because in the laboratory treatability studies were often done, but in real environment how the pollutant molecule is going to be converted to a kind of a totally non-toxic or innocuous form or not, determining those things were very important. So, eventually different type of molecular assays, not exactly the DNA or RNA based assays, but also the molecular in the sense the chemical assays, including the different type of mass spectrometry based like gas chromatography, GC-MS or HPLC-MS and other similar techniques were used to determine the fate of the pollutant, including the different other kinds of X-ray based diffraction analysis or maybe the speciation analysis etc. for the radionuclides or the heavy metal ions. Now, one of the major point with reference to this isolation and characterization of bacterial strains remain in this phase when whole genome sequencing based approaches were not implemented because they were not available perhaps. So, is that many organisms were isolated from the polluted environment. So, this is particularly true during the 1980s to 1990 early 2000 we see is a tremendous impetus was given towards isolating, characterizing organisms which are relevant for bioremediation and their possible implementation, particularly under the legacy of the US DOE, Department of Energy, United States Department of Energy, where bioremediation was attempted and successfully implemented for a large scale of sites. Large number of sites were treated using bioremediation, microbial bioremediation using specific microbial activities. So, now with respect to this isolation and characterization things, so today we are going to discuss some of the aspects before we move forward towards the genomics based events. Now, one thing that was possibly discussed in earlier classes, that oxidation of toxic pollutant remain one of the most common types of biodegradation. So, if we try to identify this process, so we have the organic pollutant molecule, so organic compound, so which is the pollutant molecule. So, it is basically subjected to the microbial catalysis. So, what microbe will do with this organic compound? The microorganism will try to oxidize it. So, they will try to oxidize it. So, So, following oxidation what will happen? Eventually, if the complete degradation is there, so carbon dioxide H2 will produce and lot of energy will be released. Now, this energy will be utilized by the microbe itself to grow and to proliferate. But for this process to happen, the microorganisms require essentially the supply of the terminal electron acceptors. Why terminal electron acceptors are required in this process? Because if we look carefully, suppose this is the bacterial cell. So, you have the organic carbon over here. So, this carbon will be oxidizing and electrons will be released. So, these electrons will be eventually transported through the electron carriers, which are considered to be the most important reducing powers will be generated, like nicotinamide, adenine dinucleotide, phosphates, etc., NADH or FADH2, etc. And finally, these electrons will be donated to the membrane bound electron carriers. So, these membrane bound electron carriers will require the terminal electron acceptor at the end. The electrons will shuttle from one into another, and finally it will go to TEA, for example oxygen or nitrate or sulphate or iron, depending upon the redox potential of the environment. So, we have discussed this thing possibly in our earlier lectures. So, one component is very important, that the degradation of this organic compound or the oxidation of this organic compound is controlled by the availability of this terminal electron acceptor. Because if sufficient electron acceptors are available and if the redox potential is favorable, then the candidate organism or the selected bacterial strain who is capable of oxidizing the organic compound, the target organic compound, will be able to oxidize it. So, the oxidation process is strongly governed by the availability of the terminal electron acceptor, which could be oxygen in case of aerobic bacteria. And so, in many cases we have found that the aerobic degradation with oxygen as terminal electron acceptor is very well studied. In many aerobic habitat or oxic habitat, surface contamination, wherever it is exposed to air, including the petroleum oil and different other hydrocarbon contaminated sites, we have found that the Pseudomonas type organism, even the Burkholderia and Pseudomonas and many similar other type of strains were plenty, and they were considered to be very capable of degrading the organic pollutants using oxygen as the terminal electron acceptor. So, you need to have the electron acceptor available, and the microbes must be capable of, they should have the appropriate enzyme coding genes within them, and other regulatory systems should be favorable. Now, in case of anoxic environment, groundwater, the underground systems, subsea habitats, wherever we have organic pollutant contamination, we see that under these anoxic environments, alternate electron acceptors, alternate electron acceptor means these are the electron acceptors like nitrate, iron, sulphate, and also the maybe manganese and other selenium, etc., could also be playing a role as alternate electron acceptor. So, eventually all these things will drive the oxidation of organic compound, which is the pollutant molecule in this case. Now, if we look at a to some extent a realistic situation where there is a surface spill, as you can see over here, there is a surface spill. So, this is some tank or something which might have leaked, and some petroleum or hydrocarbon or organic contaminant is present. It may be a landfill site also, where leachates are being produced, and these leachates are gradually moving into the subsurface. So, this entire part I will write it as this is basically subsurface, and the upper part is the surface. This part is surface and this part is the subsurface. So, with this the depth is mentioned. Now, the organic contaminant whatever has leached might have reached to the certain depth. Now, this organic contaminant would diffuse into the underground environment, and as you can see that they will possibly form a gradient, and it will distribute all around depending upon the underground geological strata or the nature of the aquifer or the groundwater flow. So, if we consider that the groundwater is actually flowing in this direction, then possibly the entire region will have this spread of organic contaminant. Now, the electron acceptors, the EAs or the terminal electron acceptors rather. So, I will write TEAs will play a very important role over here. TEAs must be present all around, but since the redox condition or the all TA may not be present also, like in the area which is closer to the surface, perhaps the concentration of oxygen will be still better, and what we can see that using oxygen as the terminal electron acceptor, the degradation of the compounds will be happening. So, you will have some degradation. But in relatively deeper region where oxygen is not able to penetrate or maybe because of other reasons the dissolved oxygen level is less. So, in this region partly anoxic condition will be started prevailing, and possibly the nitrate will help the organic carbon to be decomposed or oxidized, because nitrate reducing bacteria. So, these will be the aerobic bacteria in the first level. In the second level, we will have nitrate reducing bacteria, and so in the third level we may have the iron reducing bacteria, and that will facilitate the, so nitrate will be converted to nitrogen, Fe3 will be converted to Fe2 plus, and the organic pollutant will be degraded. In the next region it could be the sulphate which will allow the degradation, and the sulphate will be converted to sulphide, and in the next zone possibly it will be a methanogenic archaea who will use the carbon dioxide itself and will possibly allow the degradation to be happening in a very slow rate, because those archaea are not very thermodynamically is not very efficient in doing this entire business of hydrocarbon degradation, but they will work together with the sulphate reducers, as we have earlier discussed during our syntrophism class possibly. So, basically according to the availability of the terminal electron acceptors, these different zones could be possible, and in reality we have found that naturally since oxygen will always be available closer to the surface wherever the oxygen is able to diffuse, and closer to the pollutant itself, even if some oxygen is there, the aerobic organisms would consume that oxygen, because they will be trying to decompose or oxidize the organic matter, organic pollutant very quickly. So, eventually the dissolved oxygen will be depleted. So, even if you have oxygen, those oxygens will deplete, and then the anoxia will start prevailing, and under this anoxic condition this nitrate, iron, sulphate reducing bacteria will start. So, basically it is the game played by the different anaerobic organisms by virtue of the different electron transporting mechanisms that they have. Now, in this regard, what we have seen in our earlier research, particularly with respect to US DOE research, that Fe3 plus, because of its occurrence, natural occurrence in the geological strata, because it is a part of the soil system, rock system, often iron is one of the major component along with silica. So, Fe3 is one of the most abundant and also a potent electron acceptor for the degradation of the organic pollutant, because Fe3 in that case acts as the potential electron acceptor and facilitate the degradation of the organic compound. It has been found. Enhancing the availability of Fe3 in contaminated environment, organic pollutant contaminated environment, is found to be a very promising approach for microbial reduction, because that greatly stimulates the anaerobic degradation of organic pollutants. So, if we want to achieve enhanced organic pollutant degradation in an anaerobic environment, mind that in anaerobic environment where oxygen is not going to play an important role, alternate electron acceptors, of course nitrate could be there, but it has been observed that iron particularly is preferred by some of the degrading organisms. So, I will just take you to one of the best studied case, where we see that geobacter type of bacterial organisms, the bacterial strains, are found to be very important iron reducing bacteria to help in the bioremediation. Most of the US DOE or many of the US DOE sites, we have found that geobacter was found to be very important. So, initially the geobacter, the presence of the geobacter was noticed during bioremediation trial, where uranium was removed or remediated from the underground contaminated aquifer by stimulating the in situ activity of the geobacter species, because geobacter can reduce uranium 6 to uranium 4 and uranium 4 precipitates under a aquifer environment, carbonate rich aquifer environment. So, the groundwater becomes free of soluble uranium. Okay. So, from those contaminated bioremediated sites of uranium contaminated aquifers, this particular strain of what is referred as RF4T, which is basically a geobacter strain and later identified as or taxonomically designated as urani reducens, because it is capable of reducing uranium very specifically. Now, this bacterium is capable of reducing uranium as a terminal electron acceptor. So, it can utilize basically uranium as an electron acceptor, uranium 6, and uranium 6 can be reduced to a uranium 4 oxidation state, and uranium 4, as I said, will be precipitated under the carbonate aquifer environment at pH close to neutrality. And this bacterium or this strain requires organic substance. So, basically the activity of this type of geobacter bacteria can be promoted and actually has been promoted through the injection of acetate. So, acetate is found to be very useful in donating the electrons for the electron transport based the processes. And later it was found that the extracellular reduction of uranium can be possible or is possible by a conductive pili as a protective cellular mechanism, because that enables these bacterium geobacter urani reducens to transfer the electrons to insoluble deposits of iron, etc. even. And what was further achieved, that the anoxic biodegradation of different aromatic hydrocarbon containing systems can also be achieved through injecting different kind of iron reducing the iron oxides, since this Fe3 is not soluble, readily soluble, and it remains relatively less bioavailable. So, this particular work has attempted to fuse this Fe3 with some ligands, and that makes the Fe3 more soluble. So, insoluble Fe3 oxides is transformed into a kind of a binding with organic ligand, lead to a kind of a solubility, and that soluble Fe3 plus is used as the electron acceptor, while the aromatic hydrocarbons are degraded by the geobacter type of bacteria. So, geobacter is a very smart bacteria. It can degrade organic carbon present in the anaerobic environment, utilizing iron Fe3 as electron acceptor. It can also reduce uranium if some suitable other organic electron donors are there. So, you can think of electron donors like acetate, or in case the environment is simultaneously contaminated with both hydrocarbon as well as the uranium, then perhaps the geobacter would be the best candidate. And indeed, it has been found that geobacter strains are very efficient in degrading the organic pollutant, utilizing the organic pollutant as a source of carbon and electron, and on the other hand utilizing the uranium as terminal electron acceptor, thus facilitating the process of simultaneously degrading both the things. So, you can just imagine that this is the geobacter and you have the hydrocarbon, so which is the organic compound, or you might have acetate. This either of these will work. So, they will take the electrons and the carbons from these, and they will transfer these to either Fe3. So, in case of normal cases they like to transfer. So, if you have only hydrocarbon, then you can think of like hydrocarbon will transfer to this thing. So, hydrocarbon will be converted to CO2 plus H2O. But in case of uranium, you may think of that that acetate is injected, because this acetate allows the electron, because it is not a pollutant at all, and microbes like geobacter are able to utilize it, reducing uranium 6 to uranium 4 precipitates. But in case you have both this as well as this, then you no need to add, there is no need to add acetate or no need to think of any iron, because then the hydrocarbon itself will act as the electron donor and carbon source, and this will eventually go to this. So, that kind of possibilities are also there, and that is why the geobacter type of bacterial strains were found to be enormously useful. Now, it is not the case that iron is the only suitable electron acceptor. Of course, nitrate could be also another very important electron acceptor, but under strict anaerobic condition, nitrate reduction may not be feasible thermodynamically or from the redox point of view, as we discussed in our resource utilization lecture. So, sulphate is found to be another potent electron acceptor, particularly in the marine environment or the environments where we have sulphate concentration available. And if you have organic pollutants in those environment, similarly the organic pollutants can be degraded using sulphate instead of iron as terminal electron acceptor. Now, it has been also found that addition of sulphate in the groundwater is very efficient technique, because it can actually greatly enhance the biodegradation of organic compounds, because in this case the process will be the similar. So, you have this I will write SRB, sulphate reducing bacteria, and in this case the hydrocarbon contaminant will be degraded to carbon dioxide plus H2O, and the carbon and the electrons which are generated will be utilized by this bacterium, and the electrons will finally go to the membrane where you have sulphate now as a terminal electron acceptor, and you will have the sulphides produced. So, these bacterial members like the desulfobacula or desulfobacter have been found to be very important. These organisms have been found to be very important sulphate reducing bacteria which help in bioremediation. So, one thing we must be very clear about, that this degradation or the bioremediation, if it is based on oxidation process, then we should provide electron acceptor. But if it is based on reduction process, like in case of uranium we saw, then we must provide them the electron donor or the carbon source. Now, some pollutant can act as electron acceptor, as I mentioned, uranium or maybe the polychlorinated biphenyls and other chlorinated solvents. So, in that case, as I already mentioned, that the uranium or these polychlorinated biphenyl, they or the chlorate ion, they can be utilized as their electron acceptor. So, now the pollutant can be bioremediated easily. The pollutant can be bioremediated through the either the metal reducing bacteria, but like the geobacter urine reducens, or through a dechlorinating or reductive dechlorination rather by D-halococcoides. So, we will learn about the details of the mechanisms of these processes, both the metal reduction or uranium reduction and the dechlorination reactions in our subsequent lectures. Today, we are actually discussing the major progress that we have made on isolation based study. So, all these bacterial strains that I am discussing, the geobacter, the D-halococcoides, they have been isolated. They have been isolated from the contaminated sites, the bioremediated sites, and they have been investigated in terms of their abilities, that how actually they are capable of functioning. And when we were doing all these activities, that isolating the bacterial strain like geobacter or D-halococcoides or desulfobacter, sulphate reducing bacteria which can promote hydrocarbon degradation in presence of sulphate, the developments were going on in terms of the 16S ribosomal RNA gene based analysis. So, large number of sequencing events were taken up, and that type of approaches, availability of 16S RNA based approaches help us to identify the microorganisms that predominate during the bioremediation. So, possibly this has actually two implications. One is that we are possibly isolating bacteria that we are already aware of, that you have let us say a contaminated site where maybe you are trying to do some bioremediation by some existing knowledge or based on the some other information. So, you can always isolate a bacteria or a bacterium, or you can isolate many bacteria, and as a part of their identification identify them through by 16S rRNA gene sequence. So, that is always there, that you can isolate as many as bacteria possible, which might be useful for your bioremediation studies, bioremediation process development, and individually you can identify them by 16S ribosomal RNA gene sequence, that is always there. But on the other hand, you can isolate the DNA, as we have already learned, isolate the DNA from the contaminated site and then sequence the 16S. So, in earlier days, like in early 2000 we see, or maybe before that also, in many of these USDOE uranium and hydrocarbon contaminated sites, lot of attempts were made, successful attempts, and that time it was all basically clone library based analysis, because the next generation sequencing was not there. So, ideally we got 16S rRNA gene sequences. Now, these sequences can be matched with these sequences, you can always do that. So, one way is kind of a total community, I will say. So, you now see the progress. So, initially we were isolating like this part, we were just isolating many bacteria and identifying them as a potent candidate. We were, as soon as the 16S become available to us, we tried to implement that and identify them more specifically. But at the same time, as soon as the 16S ribosomal RNA gene based method was available to us, we were very clear that this can be applied directly with the contaminated site. We can actually take out the DNA from the environment and analyze. So, total community versus the isolates, we started comparing. Then somewhere or sometimes we actually tried to find out that organisms which were actually being isolated are also there in the total community. So, many of the 16S RNA gene based sequence studies indicate that these predominating microbes are closely related to the cultivable. That means, what we actually got through total community, in total community you might have some memory still with you, that some members are more abundant than others if we have a relative abundance plot. So, you have some members, member A may be more abundant when you look at the total community, relative abundance if you remember. So, relative abundance wise we may find that species A is more abundant, followed by species B and then C. Now, you may find that your cultivable species isolates are representing sometimes A itself, sometimes B, sometimes C, or sometimes even D. So, they are matching to each other. So, exactly the 16S RNA gene based analysis allow us to identify that phylogenetically many of the isolated organisms were closely related to what actually predominates, and the same thing exactly happened with the Geobacter members. In case of Geobacter, we had already isolated a number of Geobacter species, and you can understand that up to the name was given later, the Geobacter urinireducens or Geobacter metalloreducens, many Geobacter species were isolated and characterized, very thoroughly characterized for their ability to reduce metals and oxidize organic pollutants. But when we perform the 16S RNA based analysis with the community sample, we found that they are also having in reality many Geobacters, and not only it is for the Geobacter, but also for example for the trichloroethane TCE, another very important contaminant present in many of the DOE sites, the D-halococcoides type of bacteria. So, D-halococcoides another pure culture isolates were obtained, and it is not only D-halococcoides, even aerobic and anaerobic different contaminated sites like in case of naphthalene, in case of methyl tert butyl ether degrading organisms. We have isolated pure culture bacteria who are degrading these contaminants, and when we recover the or retrieve the 16S sequences from the contaminated environment, we found the same environment, we found that these isolates are actually very closely related to the members of the total community. So, indeed that is a very that is a good report or good indication that the isolation was done in a very nice manner. Now, as we progressed, the whole genome sequencing were now available. Now, whole genome sequencing of bioremediation relevant bacteria in particular, because by the time many bacterial strains including the Geobacter, Desulfovacula, Pseudomonas, Burkholderia and Dihalococoides, many strains of bacterial isolates were available, and those were subjected to whole genome sequencing. No single gene based PCR, the entire genome of those organisms were sequenced, and when we performed that, we found that it was immensely helpful in promoting the understanding on how these bacteria would function, respond and remain active during bioremediation. This was truly fascinating. So, one example I will give you, that is the Geobacter metalloreducens. So, originally before we name the Geobacter as some of the Geobacters as Geobacter uranireducens, the initial name was the Geobacter metalloreducens because it was capable of reducing metal like iron. So, originally when we isolated the scientists isolated the Geobacter strain, they found that it is actually non-motile, it is not able to move, it does not produce any flagella. But when we sequenced the whole genome, we found that the genes for the flagella, the flagellin protein are there. So, what does that mean? That means that the organism could be able to produce flagella, but the way we are testing it is not allowing it to produce the flagella. Okay. Now, the implication of the flagella is that if the bacterium is having the flagella, the bacterium will be able to move towards the nutrient, move towards the electron acceptor, move toward the electron donor. So, the applicability of the functionality of the bacterium selected bacteria will increase. Now, at the same time or the little later, the scientists investigated that if we provide insoluble electron acceptors, like iron 3 oxides. So, what was the finding? That this is the bacterium, let us say this is the Geobacter strain, and if it is provided with let us say nitrate, it will not produce any flagella. So, nitrate will give you no flagella, because nitrate itself is soluble. But if you give Fe3 for example oxides, you see flagella are produced. That means, they have now produced the flagella. Why? Because if they have flagella, Fe oxide are insoluble. So, they cannot move, Fe3 oxides cannot move, but this bacterium or the bacterial cells, they need Fe3. Why? Because Fe3 is the electron acceptor for them. So, these bacteria now will start moving and will reach to the iron dots or the iron minerals or iron particles, Fe oxide insoluble particles, and there it will stay very close to these and will be able to reduce this to Fe2 plus, and while doing this it will possibly metabolize the hydrocarbon or will metabolize acetate or other carbon sources, because it is basically a heterotrophic bacteria. So, we came to know that. The initial I have summarized the major findings over here. So, what are these summary events? Summary events are that the genome of the Geobacter metalloreducens show that it has the flagella gene. So, it can potentially produce the flagella. Then later investigations that indicate that it can produce flagella only when grown on insoluble Fe3 or Mn4 oxides, because Mn can also be used as electron acceptor. pili genes are also specifically expressed when grown with insoluble Fe3 or Mn. Now, pili is a kind of another connectors rather, which will facilitate or which facilitates the extracellular electron transfer for these kind of bacterial strains. Now, in this regard, the scientists also observed that these geobacter metallidurans and other strains, geobacter strains, they have specific advantages over other iron reducing organisms like C1Ella and Geothrix. Those are also present, but from a bioremediation point of view, we found that geobacter is more efficient. Why? Because this C1Ella and Geothrix, they produce energy expensive iron chelators, because Fe3, if they want to use Fe3 as terminal electron acceptor, they need to solubilize it. So, C1Ella and Geothrix type of bacteria, they are iron reducers, they also occupy similar contaminated sites, but they produce the Fe chelators which chelates the Fe3 and make it available to the bacterial cells. But those production of those Fe chelators are energy expensive, they need to spend lot of energy. In contrast to that, geobacter metallidurans, they do not produce any chelators. Rather, what they do, they produce the flagella and they move towards the organism directly, or they produce the pili. The pili remains a kind of a nanowire. In the later studies showed that the pili acts as a kind of a nanowire, allowing extracellular electron transfer to iron 3 molecules. Now, in the subsequent period, lot of genome sequencing of the environmental or bioremediation relevant bacterial strains, archaeal strains were performed. Now, here I am going to briefly present why do we care about bacterial genome sequencing. It is specifically to know or better characterize these organisms. You have just seen the example with geobacter metallidurans. It is also true to understand how they function as an individual or as a member of the complex association. This genomic data will allow us to understand and also to control and apply them in a improved living processes. However, the genomics is always not good, it is not enough, I will say rather, because fundamentally most microbes cannot be cultured. So, with whom you will do the genome sequencing? You should have the isolates. It was a very lucky event for those people investigating on the US DOE sites, because those scientists could isolate the geobacter strains or the D. halococcoides strain. But considering that many more bacterial strains could be there who are absolutely not cultivable, because Staley and Konopka great plate count anomaly and subsequent studies have indicated that it is close to 1% or less than 1% of the total bacterial populations are actually culturable. And also somehow we have learned over the years that culturing under different lab conditions, and if we are slightly I will not say careless, but we are not taking proper care, rather we may have lot of lab weeds which are not necessarily the dominant or most influential organisms in the environment. So, during the culturing process we need to be really very careful. It is not only the culture medium that are going to be used, but also we should take care about the other environmental conditions which might allow better recovery of cultivable bacteria. Now, as soon as cultivation independent methods and at parallel the whole genome sequencing methods are available. So, we are able to utilize these 16S RNA gene based approaches, because that allowed us to identify the phylogenetic characteristics, and what we can surely say that the microorganisms that predominate during the bioremediation are closely related to organisms that can be cultured. So, our confidence, microbiologist confidence about these bioremediation systems were built up or become stronger, and we started predicting the physiology from the phylogeny, which is basically phylogenetic interpretations were converted to the metabolic or physiological interpretations. Now, the culture independent methods for identifying the enumerating microbes were developed in a very straightforward way, because in that case the organisms they are not necessarily to be cultured, as we already studied earlier. We can extract the DNA, amplify the 16S RNA gene, and either we do manual cloning or during the next generation sequencing process, this cloning will be done through the sequencing machine or sequencing reaction itself, and then we perform the sequence analysis. Now, I will show you the basic layout, that in case of a single bacterial strain, we had a kind of only one genomic DNA, only one 16S and a sequence match will establish the identity. In case of multiple species present in a contaminated environment, we need to have some kind of bioinformatic analysis, or the cloning process should be more precise, so that we are able to segregate the 16S RNA gene sequence and analyze them. So, actually we are expecting results like this, that we will have identification at a very precise identification of the organism, like in this case a Pseudoxanthomonas strain is identified from a arsenic contaminated groundwater, and on the other case we are able to actually identify the different type of bacterial taxa which are present in a contaminated environment. But that was not the end of the road. Only 16S RNA genes are having its benefits, advantages, but also having some limitations, as we have discussed in our some of the earlier lectures. So, eventually the concept of metagenomics emerged, because that was found to be one of the best method to analyze all the organisms which are present in the environment, their functional repertoire, as well as possibly analyzing the interaction between the organisms. So, this was perfectly all right when we were trying to isolate a few bacterial strains and identifying them, but that was not about everything. We were not able to understand how they interact with themselves, how they interact with their environment, how they respond actually. All these things were not very clear to us. So, essentially the genome enabled techniques contributed to the development of models to how microbes function in the contaminated environment. So, essentially we are able to delineate the what we called as a kind of a toolbox, that the environmental samples can be subjected to a wide ranging methodology, including the isolation of the cells that could be done through the cultivation, followed by their characterization and whole genome sequencing, and then elucidation of the gene function, regulation, etc., and then developing a conceptual model for gene function regulation, and then also how they are going to function in the environment. One of the best example is the geobacter urinated cells or metalloid cells, where the organisms were isolated, they sequenced a whole genome, and then all transcriptomic and functional gene based analysis were performed to model the functioning of this organism. The alternate and parallel strategies could be the extraction of the total DNA, or the total RNA, or the mRNA, or the total protein, and we have possibly discussed part of these approaches in our earlier classes. So, eventually all these DNA, RNA, these are omics based approaches, or we can consider them as the metagenomics analysis, because here in many cases we will not be applying any PCR. It is the entire DNA or the entire mRNA pool can be sequenced, or it can be metaproteomics, where the entire pool of proteins will be analyzed, and then we will be able to obtain some inference about the function and regulation of these multiple organisms, including their interaction, their ability to interact with other species, ability to interact with their environmental factors, and then develop the conceptual model about their functionalities and their regulation, because regulation is very important over for the bioremediation process, because most of the in situ application sites or the open environments are subjected to variations, climatic variations and other variations. So, how the organism or the organisms who are the candidate organisms who are responsible for the remediation would respond, that becomes a very important task. So, what is the need of the day? So, bioremediation, whether it is a natural attenuation or engineered process, it would require a good understanding of the physicochemical characteristics of the contaminated environment, as well as a detailed description of the microbial community present. So, both are required, and it is not only true that we have to have answer of the 4 questions that a microbial ecologist often asks, but also we need to understand how different interactions between the contaminant and the organisms are playing a role. Possibly something like this, where we consider this as a bioremediation space, and both the catabolic landscape as well as the chemical landscape, where we have the nutrients to be, or the abiotic landscape like conductivity, temperature, humidity, matrix condition, etc. They control the species composition, they control the species function, and on the other hand these also will have a repercussion on that. So, in contrast to what is dominated in many studies on bioremediation or biodegradation, what we have learned that it is a case of multi-scale complexity. It may not be always true that one bacterial strain, one compound and only one pathway is going to be most important for that. It may not be. So, it is said that not amenable to the typical reductionist approach. It may not be true always. So, ideally it is the as I mentioned, it is a multi-layered structure which starts from the gene, and the genes or the enzymes, they interact with the pollutants and they also enable the pollutants to be degraded or bioremediated, but they are all organized as operons and pathways. They are part of the genomes and the cells. Then cells are the member of the species, they belong to the populations, populations belong to the community, community functions within the niche, and the niche they are in a habitat and microhabitat, and then you have the site, the polluted site where the bioremediation is to be carried out. So, when you have a pollutant present in the site, you have a number of abiotic factors. Both these pollutant abiotic factors will play roles in controlling all these parameters. All this multi-level response will be received up to the gene expression of the enzyme, which are enzymes which are going to be present, and the enzymes are going to be most important component for the carrying out the transformation reaction. So, essentially we come out with this kind of a bioremediation toolbox. What I suggest is that understanding of bioremediation space through culture independent approach would require some kind of a concept which will have a bioremediation toolbox that will include the environmental sample to be studied for multiple geochemical or environmental factors, as well as the microbiological or microbial ecology analysis, which will have both the structural and functional components. And that would possibly help us to lead us to understand the entire community function, and possibly would lead to the environmental genomics, which will answer the fundamental questions like who is there, what is done, and what is who is doing what, and then apply that knowledge into the biotechnology applications, particularly with respect to bioremediation. So, but this part of the lecture, mainly these two articles will be useful, and in conclusion, the concept of pre-genomics and post-genomics and whole genome based approaches in bioremediation are discussed. The importance of whole genome sequencing to 16S RNA gene based analysis is highlighted, and finally, we came to the concept of the multi-layered complex or complexity is introduced, and also the bioremediation toolbox is introduced with the cultivation independent approaches. Thank you so much.